Good day. I warmly welcome you all to my presentation. I am Mukti, doing PhD at Massey University. I'm working in this project as a part of my PhD. Well, the topic of my research is an investigation into the influence of age on the standardized amino acid digestibility of Australian wheat and sorghum in broilers. Amino acids are the monomers that make up protein. Amino acids can be two types, indispensable and dispensable. Indispensable amino acids are those which birds can't synthesize in their body and therefore must be supplied in the diet. Dispensable amino acids are those which birds can synthesize in their body. In practical feed formulation, we have got two options, digestible amino acids and total amino acids. Digestible amino acids is considered superior to total amino acid because digestible amino acids are literally utilized by the birds. Again, ileal amino acid digestibility can be either apparent or standardized. In apparent ileal digestibility, we don't correct for endogenous amino acid loss, but in case of standardized amino acid digestibility, we correct for the endogenous amino acid loss from various digestive secretions, pancreatic and enzymatic secretions. Here, we are studying the amino acid digestibility of wheat and sorghum, which are very different. Wheat contains non-starch polysaccharide in it, and it is a viscous grain. The water-soluble fractions of non-starch polysaccharides can negatively impact the digestion and absorption of nutrients, including amino acids. Sorghum contains different anti-nutritive factors like caffeine, phenolic compounds, and phytate. Why have we selected age? Because to date, there are few studies with sporadic data on the amino acids digestibility at different ages of broiler. At present, the amino acid digestibility values generated from older birds, mostly 22 to 35 days of age, used in feed formulation throughout the whole life. After hatchings, birds eat less. It is considered as the most critical period. Birds get nutrition from residual yolk, and yolk comprises about 20 to 25% of chick's body weight. At first week, the digestive organs like crop, ventriculus, gizzard, and small intestines are less developed. There is high demand of amino acids for organ and tissue development. After around day 10, the protein digestive enzymes like trypsin and chymotrypsin secretion and activity starts to increase. Food passage rate decrease with age. That means there is increased contact of nutrients with the intestinal wall. And another fact is that the gut bacteria, which are sterile and at first week, they become colonized and stabilized within 2 to 3 weeks of commercial broiler. And this gut bacteria has an impact on the digestibility of nutrients. There is no study investigating the standardized ileal digestibility of amino acid from hatching to end of broiler growth cycle. Another lacking is that all previous studies used mesh diets, to the author's knowledge, and in practical feed formulation, we use pellet diet for commercial broilers. And the applicability of data generated with mesh diet using in pellet diet can be questioned. And previous studies from our lab revealed that there is an impact of feed form on the amino acid digestion and different nutrient digestion of broilers. Hence, we have selected pellet diet instead of mesh diet. So the specific objective of current study is to determine the influence of age on the standardized ileal digestibility of amino acid in wheat and sorghum. Here we can see the wheat and sorghum diet which we used in our experiment. The whole study was divided into two parts. In part 1, we determined the basal endogenous amino acid loss at different ages of broiler. So, total 360-day-old broilers were divided into 6 age groups and 6 replicates. The nitrogen-free diet was fed for last 4 days prior to ileal digester collection at day 7, 14, 21, 28, 35, and 42. We can see the ileal digester collection site here. So the samples were then frozen, freeze-dried, ground, and analyzed for dry matter, titanium, nitrogen, and amino acid. Here, we can see the result of endogenous amino acid loss at different broilers' age. So the endogenous amino acid loss in case of indispensable amino acid, dispensable amino acid, and total amino acids, all are higher on day 7. And there was a decrease on the endogenous amino acid flows at day 14 to 35. There was a further decrease into the endogenous amino acid loss at day 42. To note, this study for the first time gives information on the basal endogenous amino acid losses in broilers starting from hatching to 42 days of age. Part 2 involves the determination of standardized ileal amino acid values of wheat and sorghum in broilers at different ages. This experiment was conducted with broilers from day 1 to 42 days of age. The experimental diet was based on wheat and sorghum as the sole source of amino acid and were fed for 4 days. And as we mentioned earlier, we used pelleted form of the diet. And titanium dioxide was added as an indigestible marker. And we studied 9 indispensable amino acids, including arginine, histidine, glycine, methionine, and threonine, etc. And we studied 7 dispensable amino acids like alanine, cysteine, glutamic acid, glycine, etc. We can see the ingredient composition of diets. Here, the composition of wheat and sorghum was identical, and titanium dioxide was added at the rate of 5 grams per kg. So we can see the summary of our experiment. 6 age groups were studied, and experimental diets were fed for 4 days prior to ileal digester collection. And there were 6 replicates per age group, and the numbers of birds decreased as the birds grew older, and there were total 348 birds, either for wheat or for sorghum. And on days 7, 14, 21, 28, 35, and 42 days, we euthanized the bird using sodium pentobarbital intravenously and collected the digester from lower half of ileum. The samples are then freeze-dried, ground, and analyzed for amino acid, titanium, and dry matter. Here, we can see the formula which were used for calculation of apparent ileal digestibility coefficients of the diet component, basal endogenous amino acid flows, and standardized ileal digestibility coefficients. The statistical analysis was done using general linear model procedure of SAS. Orthogonal polynomials contrast was done to see the linear and quadratic effects of age. Differences were considered significant when there was a p-value less than 5%. Here we can see the ileal digestibility coefficients of average of indispensable amino acids. The apparent ileal digestibility coefficients increased quadratically with advancing age of broiler. But in case of standardized data, there was no age influence. The dispensable amino acid digestibility, we can see the apparent values increased quadratically again, but there was no influence on the standardized ileal digestibility coefficient. Here we can see the information on individual amino acids. All right, the apparent ileal digestibility coefficients of every single amino acids increased with advancing age. We have seen either linear or quadratic effects. We can see the standardized ileal digestibility coefficients of some selected amino acids. So the standardized ileal digestibility coefficients of methionine increased as the birds grew older, and there was no age influence in case of lysine and threonine. The apparent ileal digestibility coefficients of average of total amino acids increased quadratically as the birds grew older. There was again no influence on the standardized ileal amino acid digestibility coefficients in case of average of total amino acid. In case of sorghum, the apparent ileal digestibility coefficients of indispensable amino acids were not affected, but the standardized ileal digestibility coefficients decreased linearly with advancing age of broiler. The apparent ileal digestibility coefficients of average of dispensable amino acids in case of sorghum was not affected by age, but the standardized ileal digestibility coefficients again decreased quadratically as the birds grew older. The apparent ileal digestibility coefficients of methionine, tryptophan, cysteine, and aspartic acid increased with advancing age of broiler in case of sorghum. Here we can see the selected amino acids again. So the methionine was not affected by the age, but the lysine and threonine, all the standardized ileal digestibility coefficients of lysine and threonine decreased with advancing age of broiler. We can see the difference between the standardized and apparent ileal digestibility coefficients of average of total amino acid here. So the difference between the apparent and standardized was higher on day 7 compared to all other ages. On day 14 to 35, the values were very close. It is like 10.8 to 11.5%. And there was a decrease on day 42, again 7.43%. This is same data with different graphs to see the clear picture. Here we can see the difference between the apparent and standardized is much higher on day 7 compared to other ages. So in case of sorghum, we have seen the similar trend of getting higher values on day 7 and lower values with advancing age of broiler. Again, we can see the difference here At day 7, it was much higher compared to other ages. Take home message. So, the endogenous and amino acid secretion is 2 or 3 times higher at first week compared to other ages. The application of single endogenous amino acid value for standardization can underestimate the standardized ileal digestibility coefficients in early life and overestimates in late life. It highlights the importance of correcting apparent values for age-specific endogenous amino acid losses. This study, for the first time, gives the information on the standardized ileal digestibility coefficients of amino acid in wheat and sorghum from hatching to the end of growth cycle of broilers. In a nutshell, age effect on amino acid digestibility is variable and it depends on the grain type and specific amino acid. We recommend the application of age-specific endogenous amino acid values for standardization of apparent data. In commercial poultry feed formulation in future, the age-dependent standardized ileal digestibility coefficients of amino acid may be used. To add, we have finished the laboratory analysis of 2 other grains, maize and barley, recently, and we have also finished the field experiment to see the age effect on the standardized ileal digestibility coefficients in some protein sources of broiler. I would like to acknowledge AgriFuture's Chicken Meat Program for funding this project and Evonik for amino acid analysis. Thank you all for your patience.